Hello, space travelers. It looks like you've gotten yourself into a little bit of trouble. And so you have been captured by an alien and you are trying to escape from planet Soma. Now, this alien, Oligodendrarg, um, has offered you a way off of the planet, but you must answer how each of his different modes of torture affect the nervous system. And so, in order to escape from this planet, you are going to have to be experts in the nervous system. As usual, you're going to read through this yourself, record your answers, and then I'm going to talk about the answers afterwards. And so, you're going to start by reading the introduction and scenario one. So go ahead and pause me, write that down somewhere where you can submit it to the Dropbox later, and then come back and we will discuss what this first scenario really does to the nervous system. So oligodendrog has you, and you're trying to figure out how the non-functional sodium channels affect the signaling capabilities of a neuron. And so the first question that you have to answer about this is that the voltage gated sodium channels don't open, right? And so if that happens, the neuron cannot undergo action potentials. It can go through graded potentials, and so it might reach the threshold, things might happen, but if the sodium channel is not open, it will not be propagated and it will not signal. And so the neuron that is effective will not be able to send signals. Um, Nociceptive neurons are pain receptors, and so they carry information about pain. This mutant gene might not seem all that bad at first because you won't be able to feel pain. However, in the long term, pain is a signal for the body, and it's a protective mechanism that allows you to alter your behavior so that tissue can heal and we can learn to avoid dangerous situations. If you can't feel pain, you are unable to kind of regulate your own behavior. There is a mutation that causes this phenotype, and people with this mutation have broken bones, burns, losing the end of their tongue because they've bitten it off, and lots of injuries that require like plastic surgery and other sorts of things. Children in particular have a lot of trouble with this because they can't learn what's going to be dangerous and what's not if they can't feel. And so it's important to be able to feel pain. That's scenario number one. Go ahead and read through scenario number two, and we will come back together. So this one says that it should be a piece of cake, correct? And so what we're thinking is that the um, the effect here is that the myelin sheath is going to be destroyed. Um, the action potential will travel, but it will travel much more slowly than it should, and it might even die out. The signal might die out before it reaches the axon terminal because it has such a far distance to travel. Um, this happens because the myelin sheath, what it does is it allows the signal to jump a little bit of a way. And so each sheath of myelin, it can jump from node to node, which is the space between the myelin sheaths, because it allows it to reach the terminal much more quickly. And it also acts as an insulator, which prevents the leaking of different different ions, and so it keeps the membrane potential at what it needs to be, or allows it to the signal to not die out. And so myelin's really important. Um, what would happen if you drank that orange juice? Um, it will disrupt the actions of all the pathways involved in neuron activity. And so the optic nerve carries visual signals. So you experience disturbances or loss in vision in at least part of the retina. It'll also disrupt motor pathways. And so you'll feel weak or maybe even paralysis at some point, um, at least of voluntary movement. So that one sounds like no fun. Go ahead and read through scenario 3 and then come on back. So looking at scenario 3, um, this is another one that does not seem so fun with batrachotoxin from poison dart frogs. And so the signaling will be affected because if the sodium channels open at a more negative membrane potential, the membrane will reach threshold more easily. This means that the neuron is more likely to have an action potential, right? And so when the when the threshold is lowered, then the action potential can happen more easily. This particular toxin actually lowers it by 30 to 50 millivolts. And so that means that some neurons might fire at resting membrane potential. Um, and so preventing the inactivation of sodium channels, um, it makes it so that additional sodium cannot enter the cell and allows repolarization to begin when the potassium channels open. If this inactivation is prevented, the sodium channels will remain open, allowing sodium to flood into the cell. This would at first create a burst of action potentials. Like I said, they would happen easier. But since the membrane potential is kind of destroyed by this action, it can't repolarize. It can't repolarize. And after that first burst of action potentials, there won't be able to be more. And so long term effects are pretty detrimental. Um, you'll get muscle spasms at first, including the diaphragm, which could make it really hard to breathe. And then eventually the subject would be paralyzed. Prolonged stimulation in this kind of way, if it was really easy to kind of set off action potentials, if it was stimulated for a long time, it would also deplete stores of acetylcholine, which is the messenger between our motor neurons and our muscles. And so it will block the nerve stimulation of muscles, which is really dangerous because it has similar effects on the heart as well. And so paralysis of the muscles and paralysis of the heart, that's going to lead to death. That's going to be fatal. Um, so we're not looking for poison dart frogs anytime soon. Move on to scenario 4 and then come back to me. This one is a snake venom toxin, and this blocks voltage gated potassium channels in motor neurons. And so dendrotoxin will have an effect on the signaling capability of a neuron. It blocks a type of voltage gated potassium channel at the nodes of Ranvier. And so this prevents repolarization of the membrane during an action potential. So it's very similar to the last one, but it's specifically at the nodes of Ranvier, that space between the myelin sheaths. Injection of this toxin could produce increased muscle contraction. So it would go faster, like we talked about in the last one, and tremors. And rats that have given this have actually exhibited tremors and ataxia, and repetitive spikes and bursting activity. And so victims of this bite suffer from convulsions and then eventually die, either because respiratory or cardiac failure, the muscles in their heart and their diaphragm are not working right, or because of paralysis. And so this one is an interesting one, but also very scary. A general anesthetic, what would that do? So the first question to what other type of potassium channel is, oh, sorry, go ahead and read this and write down your question, write down your answers, and then come back. So pause me now. Okay, welcome back. What other type of potassium channel is oligodendrog referring? He's referring to the potassium channels that give rise to the leak current. Um, and this contributes to the resting membrane potential by allowing potassium to leave the cell. And so it helps to keep the membrane even more negative. Um, the effect of doing this will be here. Let's scroll down here. Um, the effect of opening more of these channels will allow more potassium to flow out of the cell down its concentration gradient, which makes the inside of the cell more negative. This hyperpolarizes the cell, and if you can picture it, it's getting farther away from the threshold. And so neurons need to be more stimulated in order to reach the threshold, and therefore it's less likely to fire. Um, what will happen when the toxin reaches the reticular formation neurons that control sleep and consciousness? Um, these target neurons in the brainstem that control consciousness and respiration. Reducing the activity of those neurons will reduce your level of consciousness and your respiratory rate. And so it can become anesthesia if the concentration is high enough, but it will make you sleepy. Eventually, it will stop respiration, and so you won't be able to breathe. Go ahead, move on to scenario 6, and pause me now, and we will come back together. For this one, we're looking at increasing extracellular potassium levels to 10 times what they should be. That's a lot of extra potassium. And so increased extracellular potassium will actually depolarize the neuron and make it more likely to undergo an action potential. This will happen because the concentration gradient is reduced, and so less potassium flows out of the cell through those leak channels that we talked about in an earlier question. And the intracellular concentration is going to get higher. This makes the inside of the cell more positive, and that brings the resting potential closer to threshold. And so it is easier for an action potential to be triggered. Um, there's a type of cell that normally regulates the levels of extracellular potassium in the central nervous system. These are the astrocytes, and they actually absorb extra potassium um, because they have potassium channels in their membranes. What terror would this method produce if injected into your brain tissue? Um, it's likely to produce seizures. And so lots of extra action potentials that wouldn't normally happen. This is actually one of the things that we think cause seizure activity in some forms of epilepsy is that high potassium concentration, likely due to an absence of astrocytes. Okay, one more, and hopefully we can get off this planet. So go ahead, read scenario 7, pause me now, and come back when you're ready. Okay, so now we have sodium channels that open at more positive membrane potentials. And so this increases the threshold. And so about in reference to excitability of the neuron, increasing the threshold will require greater input, a stronger initial depolarization to the cell in order to undergo an action potential. This makes it a lot less likely that the cell will undergo an action potential, that it will propagate that signal. How does this lead to seizure activity? Seizures are produced by excess activity in groups of neurons in the brain. So that might seem like it is not so connected to this one. But this excess activity can occur when neurons receive inappropriate stimulation or when activity in inhibitory neurons is reduced. And so if an inhibitory neuron has this effect on it and its activity is reduced, then the other neurons are going to overfire. They are not going to be inhibited by that signal. And so this actually happens in some types of epilepsy where a mutant sodium channel is found in neurons that provide inhibition. Um, and so this reduction of the inhibition allows other groups of cell to be active when they really shouldn't be. And so that is how this is related to, if we go down here, how it's related to seizures. Okay, that was a lot of work, but hopefully we escaped from this alien and we are able to go home and finish our homework now. I hope that this case was enlightening about the nervous system and that you enjoyed learning about my favorite system, um, the nervous system.